All right, today we're going to go through two very important concepts that are used in biotechnology, and those two concepts are PCR, which stands for polymerase chain reaction, and also gel electrophoresis, which, if you've gone to Biolab, this is a concept that you would have touched on there, okay? And that gel electrophoresis looks like this, 50 Shades of Grey. All right, pause the video here. I'm going to give you the questions for you to have a crack on in class. All right, so in the study design, you're working on DNA manipulation still. We're still talking about the use of some enzymes. Just last time we talked about endonucleases, which are those cutting restriction enzymes. Talked about ligases. Today we're going to talk about polymerases, which we've already talked about. We know very much about RNA polymerase, okay, but we also have to know about DNA polymerase, okay? Really easy to remember which is which. RNA polymerase builds RNA. DNA polymerase builds DNA, okay? And all of these words are really related because we think of polymerase uses condensation polymerization, okay? It's really important to make those links between those words, okay? We also want to talk about the amplification, the making big of DNA using the polymerase chain reaction, and the use of gel electrophoresis to sort DNA into fragments, including the interpretation of gel runs, all right? Really important concepts in terms of forensics. All right, so let's imagine there was a murder, okay? Imagine a crime was occurred. The criminal was very careful and seemed to leave without a trace, making it very difficult for the investigators to find out who did it. But we found a tiny amount of DNA underneath the victim's fingernails. But it's so little, it can't be really used to identify the crook, right? So for example, here we can see 93% of the DNA is victim and 6.7% DNA component of tiny little quantities of things underneath the fingernail. So what do we have to do is called a process of amplifying DNA. All right, so when there's not enough DNA for scientists to manipulate, we can use a technique called the polymerase chain reaction, and this exponentially increases the strands of DNA available. This is an old PCR machine. This is a nice new PCR machine. These things on the left are really manual. These things on the right are really cool. All right, so let's go through the three main steps of PCR. Step one, we have to denature the DNA. Okay, so what we do is we heat DNA to 92 degrees for 2 minutes. Okay, and what happens is this. We have here the DNA double strand. Okay, and in between each of the nitrogenous bases, we have a hydrogen bond. And so by increasing the temperature to 92 degrees, we actually break this hydrogen bond. Let's watch it happen. Bam, easy. And they split up. Okay, so we're left with single strands of DNA. Step two is called primer annealing. We have to bring the temperature back down because we need to start reforming some hydrogen bonds. So for 2 minutes, we have the DNA at 55 degrees, and we add in those primers, and now they're able to bind, which is what anneal means, okay, to their complementary bases on each of the single strands of DNA. So, we added the primer, 55 degrees, those hydrogen bonds naturally reform. Step three involves actually synthesizing some more DNA. Okay, we're going to use we're going to use condensation polymerization, and the reason we have is at 72 degrees is because we're going to use Taq DNA polymerase, which we'll talk about in a second, and to extend those DNA strands from those primers using the normal base pairing rule. A goes with T, C goes with G. Let's watch it go. There's our DNA polymerase. There, we're going to obviously need 2 of them, 1 for each side, and there they go. They add up all of those. Now we've got 2 double strands of DNA, double the amount of DNA. So let's talk about this guy called Thermus aquaticus, which is the Taq in DNA polymerase. Thermus aquaticus, imagine what would happen if you put your DNA, if we put you, sorry, into a 72-degree tub of water, you'd probably burn. Okay, so Thermus aquaticus is a bacteria. It's a thermophilic bacteria that lives inside of hot springs like this. Okay, and what it does is that it's able to survive really well in these hot springs, which sit at about 70 degrees. So we use Thermus aquaticus because it survives so well at these temperatures, so its DNA polymerase is better able to be suited to the temperatures that we need for PCR to happen. All right, so let's do another 5 minutes to double our DNA. Let's have a crack again. So with one round of PCR, we get 2 times the amount of DNA. What about 2 rounds? Okay, we'd have 4 times the amount of DNA. 4 rounds, we would have 16 times the amount of DNA. 10 rounds, we would have 1,024 double strands of DNA. Okay, so the amount of DNA can easily be calculated using this 2 to the power of n formula. So for example, for 10 rounds, we do 2 to the power of 10, which equals 1,024. Okay? All right, so now we have enough DNA. We're going to use another technique. This other technique is called gel electrophoresis, and it can be used to sort the fragments of DNA, which are actually cut by those same types of restriction enzymes that we talked about in terms of making recombinant plasmids. Okay, as restriction enzymes have very specific recognition site, every time it finds this base pair sequence, it will cut. Okay, and that means that it will leave various different size fragments of DNA, but we're going to have lots and lots of those specific different size fragments. So this is a gel electrophoresis machine here. Okay, you have to know the different components of it. Okay, so we've got these wells at one end, and this whole blue bit is called the gel. Okay, you've got a negative electrode on one end and a positive electrode on end. DNA is always added to the negative electrode side. We connect it to a power supply, and the DNA is going to run. And this is what a finished gel electrophoresis works in a perfect world sometimes, but usually like this. No, that's not too true. Nowadays, we've got some really great gel electrophoresis equipment that will always get your DNA looking like this if you follow the correct steps. So what we do is we add all of the DNA fragments into the negative end of the gel, and that's this end here. Okay. The negative end of the gel. The DNA is negatively charged, so when the fragments move, they're going to move to the positive end of the gel. So we run a current, and because this is the negative end and DNA is negatively charged, it gets pushed away from the negative end and pulled towards the positive end. So this gel, it's a matrix of fibers which actually allows smaller fragments to move faster and larger fragments to move slower. How I like to think of it is, imagine if a monkey and a giraffe were trying to run through a forest, right? Obviously a monkey's really tiny and he's really quick. He's able to get through the forest really easy. He's going to move a lot quicker through the forest. Giraffe's got massive neck, all right? It's going to be trying to swerve around trees and under trees. It's going to take a lot longer. So bigger giraffe-sized fragments get less far than tiny monkey-sized fragments, okay? Let's watch it happen. So here we have a visualization of a gel electrophoresis. We've added the DNA to the wells on the negative side, and after time, the DNA is going to be split up based on the sizes of the fragments. The bigger fragments are going to be left at the top, and the smaller fragments are going to be left at the bottom. Okay? We visualize this DNA using UV light. Alright, so how do we analyze this gel electrophoresis? So the DNA fragments, they're compared either a DNA ladder or can be compared to other samples. Okay, each visualized band contains many DNA fragments of a specific size. So this is a DNA ladder, so this one here, and it just has a bunch of fragments, but we know the size of all of these fragments. So we can use the DNA ladder to compare the size of fragments. So for example, this fragment here I know is around 1,000 BP, means base pairs long. Base pairs. Okay, we know that this fragment is about 200 base pairs long. Okay, but we can also compare between runs of DNA. So, for example, if we have this case here, who's the dad? Well, here's the mum, right? And the mum and the child have this one the same. They have this one the same, and they have this one the same. Okay, that would leave the rest of the ones that we haven't highlighted to the dad. So this one here is the same, but this one has both of these as one the same. So this one must be the dad. So the question that I ask you is, who was the murderer?